Use of cytotoxic chemotherapies and volatile anesthetic agents may result in damage to the mucosal cells of the gastrointestinal tract. This causes enterochromaffin cells to release several neurotransmitters, including serotonin, which is also known as 5-hydroxytryptamine or 5-HT. Serotonin binds to receptors found in the cell membrane of the vagus nerve. This binding activity triggers signals to the brain that result in nausea and vomiting. One event closely associated with this signaling network is serotonin-induced calcium influx through the 5-HT3 receptor, a cell surface ion channel composed of 5 subunits. 5-HT3 receptor antagonists are drugs that inhibit the effects of serotonin on the receptor and block calcium influx. Older 5-HT3 receptor antagonists are agents with a chemical structure similar to serotonin. They are believed to compete directly with serotonin for this same binding site on the 5-HT3 receptor. These competitive binding properties block access of serotonin to one or more receptor sites, thus diminishing calcium influx. Older 5-HT3 receptor antagonists exhibit simple bimolecular binding and no cooperativity. This means that an antagonist binding at one receptor site is independent and does not influence additional binding at another site. Research has begun to demonstrate that the molecular properties of 5-HT3 receptor antagonists may not be identical. Palonosetron is believed to be different from older 5-HT3 receptor antagonists in several ways, starting with its plasma half-life of approximately 40 hours. This is considerably longer than the half-life of other antagonists. Palonosetron also exhibits binding affinity for the 5-HT3 receptor that is 30 to 100 times greater than older 5-HT3 antagonists, such as granisetron and ondansetron. In addition, the chemical structure of palonosetron does not resemble serotonin or older 5-HT3 receptor antagonists, suggesting possible molecular interactions that could be unique to palonosetron. And unlike other 5-HT3 receptor antagonists, palonosetron exhibits allosteric binding, meaning palonosetron binds to the serotonin binding site in addition to a second or allosteric site. Binding to an allosteric site causes a conformational change in the receptor that increases the overall affinity of the receptor for palonosetron. This phenomenon is called positive cooperativity. Positive cooperativity makes it more difficult for the natural ligand, serotonin, to bind to the receptor, even at high concentrations. As a result, calcium is unable to enter the cell. In addition to blocking serotonin binding, the allosteric properties of palonosetron help accelerate dissociation of older 5-HT3 antagonists that are already bound to the receptor. Because of preferential binding of palonosetron, it becomes harder to displace from the receptor site, resulting in a long-lasting inhibition of 5-HT3 receptor function. A characteristic unique to palonosetron is the ability to trigger 5-HT3 receptor internalization. While it has been demonstrated that agonist-triggered 5-HT3 receptor internalization occurs in response to serotonin, palonosetron is the only receptor antagonist to demonstrate this ability. It is hypothesized that palonosetron binds to the allosteric site and causes a conformational change that facilitates receptor internalization. As a consequence, fewer 5-HT3 receptors are available for serotonin binding at the cell surface. This process appears to take place early after palonosetron exposure. Due to its great affinity for the 5-HT3 receptor, palonosetron would be expected to stay bound to the receptor for an extended time upon internalization. After exposure to palonosetron, the internalized receptor may remain inside the cell or be targeted for degradation. Older 5-HT3 receptor antagonists, such as ondansetron and granisetron, have minimal to no effects on receptor internalization. Rather, they compete for ligand binding to the receptor at the cell surface, which can be displaced by circulating serotonin. With little to no receptor internalization, these older 5-HT3 receptor antagonists do not demonstrate prolonged receptor function inhibition. In summary, palonosetron has a longer plasma half-life and potent binding affinity compared to older 5-HT3 receptor antagonists. Palonosetron also exhibits allosteric interactions and positive cooperativity, triggers receptor internalization, and exhibits prolonged inhibition of receptor function. These results may provide a pharmacological basis for differences noted in the control of acute and delayed nausea and vomiting in clinical trials, comparing palonosetron to other 5-HT3 receptor antagonists. All these properties combine to differentiate palonosetron molecularly within the 5-HT3 receptor antagonist class.